Here's a little bit about promoter and mapping the location of transcription onto DNA. So here's my DNA sequence. This is duplex. The promoter is actually a DNA sequence found in multiple places in bacteria where RNA polymerase binds to start transcription. The promoter has several regions. The first one, first of all, I should state that the start of transcription is termed +1. It always starts at +1, and it's usually at a purine. Now, orientation in this direction, I guess first of all I should say that transcription is going to go in this direction. We generally represent it going from left to right. So if transcription is going from left to right, we say that this is upstream from the start, and this way is downstream. Locations that are upstream have negative numbers, and it's the number of bases upstream from +1, and downstream has positive numbers. Our promoter is this region of DNA, actually including the start site. So the promoter is DNA upstream and including the start site of transcription. The most common parts of it are +1, and then also there is what's known as the Pribnow box. And that is located 10 bases upstream from the start of transcription. There's also a -35 region. The Pribnow box has a consensus sequence of TA, TAAT. Consensus. This is a term that you'll hear a lot in biochemistry, and what it means is those are the bases that most commonly occur in those positions. There's a -35 region, which has a different sequence, and then further in that direction, there's also something known as an up element, which doesn't always occur. And you'll see in more detail on the next slide. So this is showing the promoter sequences from various genes. These are the names of the genes in E. coli. So here is the start of transcription. It's either CAT or CGT. Those are the consensus sequences. And here's the Pribnow box in the -10 region with consensus sequence TAT. And those numbers are the percentages that those nucleotides occur in that position. The -35 region is shown here. Notice that these sequences are rich in AT base pairs, and that allows for strand separation, which has to happen in order for the transcription bubble to form. We have to have the double-stranded DNA separate so that RNA polymerase can base pair with the template strand. It's also worth pointing out that this is the consensus sequence, and even though this is not the template strand by convention, it gives us the 5' to 3' or the sense sequence, even though the template runs from 3' to 5'.